just joining us, welcome. This is SynBioBeta Live, and my name is John Cumbers. I'm the founder and the CEO of SynBioBeta. And today we're talking all about environmental sequencing of the COVID-19 virus, that is the SARS-CoV-2 virus. And we have three very special guests joining us today. We have Josh Perfetto, who is the CEO and the founder of qBio down in Santa Clara. We have Emily Leproust, who is the CEO and founder of Twist Bioscience here in San Francisco. And
Hello.
We have Chris Mason, who is the Cornell Weill professor at Cornell Medical School. Welcome, Chris.
Hello.
So thank you all for joining me. We've got some very exciting work that everybody's doing in terms of environmental sequencing of the COVID-19 virus, which is the SARS-CoV-2. And we're going to go through a few slides that each of our panelists have put together to help us understand what they're doing in this area. And then we're going to have an open Q&A with everybody, and we welcome your Q&A. So if you're just joining us and you didn't hear me earlier, type your Q&A into the question box, answer other people's questions if you know the answer, and upvote the questions that you want me to ask. In addition, we want to know where you are dialing in from today, and we can see a lot of people in the chat box. We've got people from BASF, Tanya Jackson. Hi, Tanya. Long time no see. Thank you for joining us. We have people from North Carolina. We have from Edinburgh, Scotland. We have Boulder, Colorado. Hi, Larry. Good to hear from you. Somebody from Samsung dialing in. Ellen Jorgensen. Somebody from Vienna in Austria. Welcome, Roland. So we have a great group joining in. Khalid from STEMloop at Northwestern University in Chicago. Welcome. Great to see so many people online. So as I said, we are live, and if you want to ask questions about what's going on today and what we're doing in the chat box, in the Q&A box, then please do. So without further ado, I want to introduce you to all of these people are my good friends. I was just talking about how long we've known everybody, and it's between 6 and 10 years that I've known all of our three panelists today. So it's really wonderful to have them all together on this town hall today. Josh, I've known because he was one of the founders of BioCurious, the biohacker space in Sunnyvale. He was also the instigator of the Open PCR project, the Open qPCR project, and then the founder of qBio. And Josh, if you could say a little bit about, for people who are just joining us, about what qi does and some of the application areas. And while you're doing that, we're just going to put up a poll today to ask the audience's expertise in the area of environmental sequencing, whether you're familiar with qPCR, whether you're familiar with viral sampling, what your expertise level is, and that's going to help us pitch how we explain today's content. So if you can ask that in the poll, I see it's coming up on the screen, and then while we're doing the presentations, that's going to help our panelists understand how to pitch it. So Josh, over to you. What is give just a very brief explanation of qPCR and what it is, and then what qi is doing.
Yeah. So qi is really focused on making molecular biological technologies more accessible. We started about 5 years ago when we did a Kickstarter for the Open qPCR device. And so qPCR is a way that you can detect any kind of DNA or RNA sequence. So very useful for detecting microbes as well as viruses, such as the current COVID-19. So we focus on making it lower cost and easier to use to make it accessible. So for example, we have people using this in food safety applications, in breweries where they look for beer spoilage organisms, water quality, all sorts of, basically everything under the sun.
So in the environment all around us, we know there's a lot of microbes. The microbes far outnumber other organisms. Even in our bodies, microbes far outnumber other organisms. And there's also then a lot of viruses, because these viruses are getting into our bodies, they're getting into these microbes' bodies. So why is it important to track these down in industrial situations, and how is it done?
Oh yeah, there's a lot of industrial uses. So like in food, we're really concerned about Salmonella, Listeria, like E. coli. And it's done kind of two ways. One is that we test the food, but the other is that we do a lot of environmental testing. You can't test every piece of food to make it safer. You'll destroy all your food testing it. So you do a lot of swabbing of the environment to try to look where is like Listeria present, where is it harboring when you're doing your cleaning, in little cracks and stuff like that. So this is a very well accepted technique in food safety.
And the point is that by the time that the E. coli gets into your lettuce at Whole Foods, it's too late. So you want to try to get to it earlier. So you want to, for example, if you're running an E. coli farm, you might want to be swabbing the packaging plant for the E. coli or the plastic bags that you're putting the lettuce in before it gets to the stage of getting contaminated on the truck or getting to Whole Foods. Is that right?
Yeah, yeah, absolutely. I mean, you want to be more proactive, especially if you've got a processing facility where a lot of food is going by. If you get some contamination, especially salmonella, a whole bunch of food can get contaminated. And really, you have to clean, and that's the only, to ensure that the food is safe. But it's really hard to detect where it's harboring, and so that's really why we do the environmental testing.
And most of it that's going on in the environment in terms of food and industry is mostly the things that you're mentioning, like salmonella and E. coli. These are bacterial cells. But is there much need for testing viral contamination in industry and in food applications?
Yeah, so that's not really as commonly done. I mean, norovirus is a concern, but yeah, most of the testing that we do is bacteriological. I think what we're seeing with this, COVID-19 outbreak is that this is really one of the first times we start to see a lot of interest in virus environmental sampling.
It's a golden age for biology. Walter Isaacson, the biographer of Steve Jobs, had a wonderful piece in the Wall Street Journal yesterday that you should check out if you haven't seen it, all about the golden age of biology and how things like what SynBioBeta is doing in SynBioBeta Live and bringing science out of the closet and into people's homes is going to see a flourishing of people's understanding. Even my own understanding of viruses and viral genomics and immunity has just shot up just in the last 2 weeks from doing these. So very interesting. I'll let you get onto the slides in just 1 second, but before I do, tell us about the beer. That's what everybody's that's on everybody's lips right now. So one of your big customers is the home, not the home brew industry, the craft brew industry. Why do they care about what's in their facilities?
Yeah, that's a very interesting question. And so like actually brewing is kind of a very basic biotechnological application where you're using yeast to ferment this. They have two types of problems. One is like lactic acid bacteria that can spoil the beer and make it go sour. So it could be like fine when you package it, but over time as it goes through the distribution channels, these things will grow and sour it. The other thing you have is wild yeast. There's like something called a diastaticus, which basically can like over ferment the beer, and it will make it actually continue to ferment while it's in that bottle or the can. It not only affects the taste, but it actually causes this overcarbonation, which can make the whole thing explode, which is very bad for the consumer, very bad for the brewery. So this is a very basic QC that all breweries have to do to ensure good quality.
Here's to that. Great. So the poll results are back in. We see that about 50% are intermediate, about 40% beginner, and 16% advanced. So that'll help us to pitch where we hit this conversation, right down the middle. And we have 43% of you joining us from industry. So the majority of people from industry. So that's great. Thank you, everybody. So Josh, if you want to just share your screen. And we have a new policy on SynBioBeta, which is no boring slides. So I've asked all the panelists for no boring slides. If there are boring slides, you're going to hear a giant sound of a klaxon going off, and I'm going to ask you to move on. So Josh, without further ado, over to you.
All right, great. So, oops, sorry. So here at Chai, we really have been thinking a lot about what do we do kind of between now and when we are able to kind of resolve this problem of COVID-19. So just a few days ago, SynBioBeta sent out something, an infographic of like when antibody treatments were going to come online, and that was expected to be probably August this year, and vaccines going into kind of late next year. And so we're thinking about how do we get the economy more opened up between now and when we have that.
And if you just want to hit present, Josh, you'll be in the screen.
Yeah, okay, here we go.
Thank you. Oh, you hit present a view, I think, instead of present.
Shit. Oh, sorry. Okay, sorry about that. The Zoom thing has come up over my normal thing here, so I'm trying to.
Yeah, just down there on the left, the little present down beneath the apply to all, I think.
Let me maybe just make this a little bit bigger. We'll just go through that.
Yeah, it's all good. Just go for it.
Okay, sorry about that, guys. Broke the rule of no boring slides. So anyways, kind of the SARS-CoV-2 has been challenging because people can be contagious before that they have the symptoms. It's up to a 2-week incubation period for the symptoms to occur. Not everybody goes and gets tested because they have very light symptoms. And so we all kind of seen that the net result is that a number of people have been exposed by the time a positive case is detected. And so we're trying to think about, how do we use what I was just talking about we do in the food safety industry for environmental testing to detect the SARS-CoV-2 earlier. So what we've been doing is looking at testing something called fomites, is the technical word for this. A fomite is basically some sort of inanimate object where people get can get a viral infection from. So we've all been told, cover our coughs, don't wash our hands, don't touch our face. And that's because people can, really infect their hand by sneezing on it, touch things like door handles and contaminate that. Other people touch that door handle, contaminate themselves, and then kind of touch their face and get contaminated. So that's a problem, but we can use it as an opportunity, to swab these surfaces, like door handles, pin pads, any sort of high touch surface, bathroom doors, handrails, and then use that to detect the presence of the SARS-CoV-2 in the environment. And that, if you're able to do that, you can then take mitigating actions. And an obvious one from the food industry is that we do increased sanitation and cleaning, but we can also use it for public health purposes. Initiate more selective and surgical social distancing, or what facilities need to be closed. I think we can also use it the opposite way as well. Kind of show where the virus is and isn't, and maybe where there can be green zones where there can be less social distancing. And then I think another really important application is recommend diagnostic testing. If you think about, like, you have an internal door in a facility that's only open to employees, and inside of that internal bathroom you detect this virus, then that's the indication that maybe one of your employees is infected, and they could be recommended to seek out diagnostics testing due to a potential exposure.
Excellent.
What we want to do.
Yeah, this concept of green zoning we can come into in the Q&A, and that's going to be interesting when we bring in Chris Mason to talk about what he's doing in New York. So the qPCR is the solution that you have. So talk us through that quickly.
Yeah, so we have like we tried to make this very easy. So we have like a 3-step process where basically people will first swab the sample. Then we add this to a tube where we do a 1-step RNA extraction. This actually uses the same RNA extraction system that we use in our COVID-19 laboratory test to make it really easy and high throughput. And then the result is put into this qPCR thermocycler, which is the same RT-qPCR technology that's used by the diagnostic labs to do this testing. So we really wanted to make it very easy. We have starter kits like this that let all sorts of businesses do this. It's the same type of solution that we've done in things like breweries.
Great. And we have a question that's come in. Is qPCR reporting RNA or DNA or SARS-CoV-2 for SARS-CoV-2? And what are your thoughts regarding ineffectivity of the organisms reported by qPCR? I guess I'm not quite sure I understand. Maybe it's talking about false positives of viral DNA.
Yeah, so the technology is called reverse transcription qPCR. So the reverse transcription is we first take the RNA and go to DNA, then we do the qPCR, which like exponentially amplifies or copies the DNA to the point that the instrument can detect it. So the selectivity of this technology is very good. That comes down to the what's called the primer and probe sequences. And so there's really there's always a risk of false positives, but I think, in my opinion, that's very low. I think the bigger thing is the limit of detection. How sensitive is this? Because if we detect it, that shows something is present. But if we don't detect it, it doesn't definitively say it's not present.
Got it.
And that's the big question.
Great, excellent. Well, thank you very much, Josh. We'll come back to you in the Q&A, and I know you've got some interesting thoughts and discussions that you'd like to have. As a reminder, this is a live town hall, and we'd like you to ask your questions in the question box, the Q&A box, and we'll also be taking live Q&A from the audience. So if you would like to speak to our panelists, please raise your hand in Zoom and I will call on you. Make sure you've typed in your question in the Q&A and then raise your hand and you can ask your question live. Otherwise, I'm happy to ask it for you. Next up, we have Emily Leproust. As I said, Emily is the founder and the CEO of Twist Biosciences. It's a publicly listed company that is making DNA. Emily, where are you right now?
Good morning, John. I've been grounded for a few weeks, so I am in my happy place. I'm in United Economy Plus. But if you want to be serious, I can upgrade.
Yeah,
and,
I think that
and go to the ISS.
Okay, excellent. And that's going to be a wonderful segue into Chris Mason shortly. So wonderful. So Emily, Twist is making DNA. Tell us a little bit about the DNA that you're making, and in particular, some of the things that you're doing around this response to COVID-19.
Yeah, thank you. So at Twist, we've built a platform based on silicon to write DNA. So the chemistry of writing DNA is known. You can buy a bottle of A, a bottle of G, a bottle of C, and a bottle of T, and you can build any piece of DNA that you want. And there's a number of applications for that. One of them is in diagnostic to understand the genetic makeup of a sample, and I can share a slide on that if you want.
Yeah, please do.
Yeah, so what we discussed earlier with Josh is an RT-PCR test where you get a yes or no answer, but which is very important. And the next step is you may want to know what exactly is the virus. Are there any mutations in that virus compared to what is known? And so you may have a sample, can be a patient sample, can be an environmental sample, and in that sample there is a mix of the virus RNA or cDNA in this case in red, but it's in the sea of a lot of other nucleic acid. And if you sequence that, it may be hundreds of dollars or thousands of dollars to get to what you really want, which is the virus. And so at Twist, we use the DNA that we make on our silicon chip as magnet, and we basically can extract, it's called target enrichment, you can extract just the RNA sequence out of that sea of other nucleic acid, and then you can sequence it. And when you sequence it, it's not just a yes or no answer, which is very important, but it's the next step, which is what exactly is the virus. You get a read of every base of the virus. And here's a typical readout. And in this case, we've compared the Wuhan coronavirus versus the Australian coronavirus. And there's a 10 base deletion. There's 10 bases that are missing in the Australian version of the virus. And I'm not showing it on this slide, but somewhere else on the virus, there is another 3 bases that are different between Wuhan and Australia. But now you can do that at scale. You can compare Italy and New York and San Francisco. You can even look at every different patient to see how the virus is evolving and inform policy.
Excellent. Well, thank you for that explanation, Emily. So just to repeat for everybody at home, when you're sequencing, particularly when you're sequencing environmental DNA samples, you've got a lot of stuff in there. You've got my DNA, you've got the bacterial DNA that might be in the environment, and then you've got the viral DNA. So you've got to, if you just went and sequenced everything, you'd just you'd spend all your money sequencing everything, and you wouldn't maybe be able to get to the answer that you're looking for from the question that you're trying to answer, which in this case is how much COVID-19 viral DNA is in my sample. So what Twist offers is what's called a probe panel. Is that correct?
That's right. We offer the magnet in a way, and those are exactly our DNA probes that have a biotin on it, and that DNA probe will go and capture through hybridization. They will capture the virus sequences, and then you use the biotin and streptavidin bead to do a physical separation of the virus from everything else.
So these are 2 molecules, biotin and streptavidin. These are 2 that have a binding property. They like to stick to each other. And so you've got one of them that's in the viral sample, that one of them that's in the viral sequence that you're looking for. So then that allows you to stick the magnet in with the other sticky molecule on it, and it pulls out everything that you want. And then you can wash it off and send it to sequencing. Is that right?
That's right. And then so what it looks like, what it looks like, here we did an experiment where at Twist we did not have access to the virus, so we just made it from scratch. So we made synthetic controls, and we did
virus. I should add not the.
Yeah, it's not live. It's the virus is cut in 6 fragments, so there's no biosafety issue. It's safe. You can brush your teeth with it, and then we did titration experiments where in multiple samples we use different levels of different amount of virus, as high as 1 million copies and as low as 1 copy. And so if you look at theoretically, what's the viral fraction? It's very, very small. It's a very, very small amount of the virus in the sample. And so if you had to sequence the whole thing to get to the virus, again, it would be an enormous cost, hundreds of thousands of dollars, which you can do once in a while, but you cannot screen an entire population. And then after enrichment on the right, you can see that this is the result for 1 million reads on the sequencer. Even if you only have one copy of the virus, you still get 300 reads from the virus, which is a viral fraction of 0.03%. And so it's hundreds of thousands of fold enrichment. And so that means that you have to spend hundreds of thousands less than if you sequence the whole thing. And then obviously, if you have 1 million copy of the virus, almost all of the reads are from the virus. So you can save tremendous amount of money, which means that you can deploy the technology to many more samples, and now you can sequence every human and every doorknob that you're worried about.
Great, excellent. Thank you, Emily. And can you explain a little bit how these work? Your company sells these panels of DNA for enriching so that you don't spend all your money on sequencing everything, but you spend your money on sequencing what you want. If I'm a craft brewer, like Josh was explaining earlier, or I'm looking in my lettuce factory for E. coli, can I make a specific panel that's going to search for things that I'm looking for that are contaminating my sample? Do I buy these set things from you? Like, do you have a panel specifically for COVID-19 virus and another one maybe for listeria for food sampling? Or do I have to go and tell you exactly what I want in my panel and then you make it for me?
Yeah, so the answer is all of the above. So we have a pan-viral panel. So it's on the shelf that will look for all the viruses known to mankind. Once if you identify a specific pathogen, then we can build it from scratch for you. We can make a synthetic control, and then we can make a very specific sequencing NGS panel just for what you're looking for. So we have one for SARS-CoV-2. We have one for respiratory panel. We have one for the entire virus of the entire family of coronaviruses. But then it can be very custom. If you have your own particular nucleic acid genome that you want to look for, you tell us, and we'll make that custom for you. So it's really whatever you want. We have some on the shelf, and then we can make any as a custom synthesis. Fantastic. And that's the power of being able to write DNA at scale very affordably, is that you're not stuck with whatever is on the shelf. You can really customize your science to what's needed for you.
Fantastic. And Emily, you talked about cost. What is the cost of one of these panels, roughly?
Yeah, and so when we did an experiment, the cost of the sequencing to get something like that can be as low as $5 per sample for the sequencing. And there's a little bit more for the reagents on top of it. But the sequencing portion can be very affordable, such that you can really push it to as many samples as you want.
Great, excellent. So we're going to come on to Chris Mason very shortly to talk about swabbing the New York subway and what's going on in that regard. But Emily, if you can just keep up your slide, because I do want to talk about some of the other work that Twist is doing in the biopharma space.
Sure.
Because it's related to the infographic that we put out just yesterday with Leaps by Bayer, particularly around two of the therapies that are coming in timelines towards this. Twist is also doing some very exciting work both in the vaccine development and in the development of antibody therapeutics. So Emily, I'm going to let you just say a little bit about both of those. But before we do, I just want to remind everybody that this is a live town hall and we want to hear your questions. I see that we have 9 questions. I'd like you to help me decide which questions we want to ask the panelists. We'll get through as many as we can. So just upvote the questions that you want me to ask. And we'd also like you to ask them live. So if you've asked the question and you want to ask it live, just raise your hand and we'll call on you as soon as we've gone through all of these panelists here. So with that, Emily, tell us a little bit about the work that Twist is doing both on the vaccine front and the antibody therapeutics front.
Yeah, thank you. So what we've talked so far is about the reading part of DNA, which is very important. As Josh mentioned, if you can read, you kind of know what to do. You can wash more. You can isolate socially, which is good. But in addition, if you can write DNA, now you can have even more proactive action. And there's two threads that we are supporting. One is vaccine. And you have companies like Inovio who get infectious part of the virus from Twist. We write those attenuated sequences from scratch, and then they do the work of developing a vaccine from that. And so then the idea is in 14 months you may have a vaccine that will protect everybody, or will create, if not everybody, at least create some herd protection, so that there you need access to a lot of DNA to try all of those different vaccine ideas. And then the second thing that we can do is around therapeutic antibody, where it could be that some people don't respond to a vaccine. They may still get sick, and so there it would be ideal to have a drug on the shelf that will neutralize the virus. And so what happens is there's already a few people that got the virus and they recovered, meaning that their immune system has mounted a defense. And so those people in their body, they have an antibody that just neutralize that virus. And so groups like the Crow Lab at Vanderbilt University got access to those patients that recovered. They sequenced the immune system of those patients, and they got a list of potential antibodies. They don't always know which one is the one. And so what they've done is they've ordered from Twist thousands of those potential antibodies that we're making. They will screen them, and then hopefully they will find the one that they'll be able to make at scale and use as therapy for the future.
And I'm just bringing up our infographic that we published yesterday. If you haven't seen it, you can go online and download it. We actually are encouraging people to take it and adapt it for their own use, as long as you keep the logos of SynBioBeta and LeapsByBeta on it. We're happy for you to do that, and we'd love you to do that. We've actually had it translated into Spanish yesterday, which was wonderful. And you can see on this infographic, this is what Emily's talking about right now, which is the antibodies from the patients who have recovered. And that's a big hot area. And we'll be hopefully having Jim Crow from Vanderbilt on the show the week after next. And there's also other models that are going on in terms of producing these human antibodies in mice, and then engineering and evolving these from previous vaccines that have been developed. And then once they get inside the cell, that's how they're active. Emily, thank you so much. That's a great explanation, and I appreciate it. Chris Mason, over to you. I'm going to stop sharing my screen, and the segue, of course, from Emily, with Emily on the space station, is that Chris, you're pretty famous for sequencing astronaut poop. Tell me about that.
Yes. Well, and you can hear me okay. The audio sounds good.
We got you.
Great.
Yeah.
I'm a big fan of stool in all its forms and places, including space. Not for every instance in use, but at least for the medical and sort of diagnostic utility, it's very clear. So we published last year the twin study. Most people have probably heard about that, but it is a complete multi-omic longitudinal examination of what happens to the human body in space, and also looking at sort of the walls of the space station. We have several papers in review about that. So there's everywhere we go is a living ecosystem. We're kind of all in this big nucleic acid soup together. So that gets kind of a segue into what other work we're doing in the lab. Yeah.
And just very briefly, this was the astronaut twins. One of them was on the space station for a number of months, and then the other one was on Earth. And they had their microbiome sequenced, all the bacteria inside of their gut, before they went on the space station and after they went on the space station. And just very briefly, Chris, what were the differences that were found from that study?
Yeah, so we actually examined oral microbiome, skin microbiome, stool, also see what happened in actually their gut diversity in terms of what was found in stool. It actually, did stay relatively stable, but the ratio of different species, such as firmicutes to bacteroides, did shift in a potentially negative direction, but nothing that was clinically or medically dangerous. But it did get disrupted, you can see, in flight, and then came back down. Whereas, Mark Kelly, who stayed on Earth, who's now running actually for Senate in Arizona, his microbiome stayed much more stable. So you can see there is a bit of a perturbation of being in space. And we see the same thing with mouse models as well when we fly them up in space. And they kind of float around in the mouse cages, unsure what to do for a while.
Great. Do you endorse that microbiome?
Endorse the microbiome, like to use it, if it's useful, yes, I'd say yes.
I was just trying to make a clever joke about him running for space. Let's
Well,
move on. If you want to open up your
yeah,
go ahead.
Scott Kelly did write an op-ed in New York Times because he said, okay, everyone's staying at home. Let me give you some advice. I had to stay in one tube of aluminum for a year. Here's my advice. So it was a really great op-ed if you want to know like how do you survive being holed up in your house for a while. There's a good tutorial about it.
Chris, are you friends with Mark?
Email, and we're still doing testing, so we chat occasionally whenever there's exciting results. But we haven't sat down and had beers together or anything quite intimate. But it's been it was still, the study in some ways is still ongoing. So yeah.
When are you going to have results coming out, and can we get Mark on the show?
Yeah, hopefully. Mark Orscott there. So there was one big paper came out that was across all teams, but there's still another about 12 different papers, all in various stages of production that detail more immunological changes, other microbiome data, looking at transcriptional dynamics, microRNAs, other sort of vagaries of the telomere sequencing. We saw strange things. So there is a lot more papers coming out soon. So it might be timely in a few months if some of them come out to have them out back on the show. It'd be a great idea.
Fantastic. Well, we have a lot of questions coming in, and we'll get to the questions shortly. You can vote the questions up or down, and you can raise your hand to ask them to our panelists live. So, Chris, everybody's waiting to find out what you've been doing on the New York subway. You've been going around there, swabbing the subway, and finding interesting viral sequences and other sequences. So, this isn't your first rodeo at doing this. Tell us about the work you've done in the past, and maybe you want to show us some of the pictures of what's been going on in New York.
Yep. And I've got a few pictures of some swabbing that we've been doing and preliminary results of what we found in the subway system. So I'll start off with a bit of good news. Is we don't have any good solid evidence that there's SARS-CoV-2 in the subway. We just have a very limited data set so far, but the collection is ramped up and we're collecting lots and lots of samples. So I'll show, we're going to post a lot of this data into a preprint that I was hoping to be online by today, but it's probably going to be on early next week. And I'll just jump right in. So we've been swabbing, as some of you may have heard me tell the story, I got really interested in metagenomics for two reasons. One is I kept sequencing human genomes, and not all of the reads that were supposed to go to human landed there. It's because they're microbial. So even in tumors recently, there's been a lot of papers showing microbes can be embedded in tumors and change how you view chemotherapy. So there really is a much more appreciative cross-kingdom view of biology, both in my lab, I think in the field in general. So that was one thing. The second thing is when my daughter got old enough to ride the subway, she did one day actually lick a subway pole, which I was terrified about. So I really wanted to know what was there, what had happened. There had been, we all have microbial exchanges, with our mouths when we're kids, and that's part of growing up. And some people, when they're in middle school, it's a longer story. But there is, a continual microbial exchange that happens. It's just not usually in that way with the subway. But I just got curious. And so we've been swabbing the New York City subway since 2013. And now globally, many other cities. And I have a quick few slides I'll go through. Let me quickly share my screen, and I'll kind of show how this links to some other features for other work in the lab. So in particular, as most of you know, I don't have to belabor the point, there's a lot of cases around the world right now, and the US just eclipsed China as the place with the most cases. So what we've been wanting to do is leverage a global network of science that we have here. So we're doing the New York City subway, but we are one of a large phalanx of researchers and scientists who want to do this sort of swabbing. Let's see if I can't quite hide it perfectly, but you get the sense. This is the MetaSub Consortium, which is metagenomics of subways and urban biomes and seeing what is happening in our city. Every summer we do a sampling, but we've moved things up this year to do 2 samplings because we want to actually see what's present on the RNA of different surfaces. So in this case, one thing, just a quick note about the MetaSub Consortium, they're very dedicated scientists. You can see here, wow. last year, Alina, she actually got arrested, but then before she got sort of detainment, she said, can I swab the detainment cell? So she did that. Some people get very creative in their annual sampling. This is Nuranan Najrahan's team in Singapore. So it's really great stuff on Twitter that comes up every summer. It's a really fun group of scientists to work with. I have the pleasure to work with. I see some of them on the call here today. Bharat, for example.
Was she really arrested, or were you just joking?
Well, detained. Arrested is strong. She was detained briefly, but was let go. So it was just for a little while.
What was she detained for?
Well, some people get upset. So the biggest danger of swabbing in public places is that Some people think correctly that you're just collecting a sample. Other people, however, think you might be leaving something behind, which it can be technically a crime if you do it, if you do it the wrong way. So, but keep in mind, we all leave our skin cells everywhere we go. So in some ways, you have no choice. But this is a couple of pictures from last summer. But what we've been doing, starting about 2 months ago, seeing the pandemic emerge, is we started with the pilot cities, and it's expanded out just this week to do almost all the rest of the cities. But these are the ones that are out swabbing and collecting RNA samples, and you can see here the cities listed on the map. So far, it's using an isohelix swab and putting in a DNA RNA shield, so it preserves the RNA at room temperature for at least 30 days. The pilot was 1,600. We're going to expand out for the whole tracking. It's about 10,000 samples. That's ongoing right now. And the data, the consortium is very open. So if people want to see the data, we're uploading some of our current RNA sequencing data up onto a Wasabi portal so people can play with the data. The principles of it are very much about open science and sharing methods and best practices. And so it's especially relevant for times like this when there's a global crisis, so we can all work together, share reagents, share expertise. Just a couple of pictures from the SARS sampling effort that we have ongoing. Here's David Denko from the lab doing floors and air sampling. You can see here, this is near Penn Station. This is from Seattle, Washington. You can see there's not that many people left as of a few days ago out and about. In the Alexanderplatz, this is in Berlin, so you can see some of the other places here. And then also Argentina. So just kind of a quick snapshot of different places. South Korea, Daegu, which is one of the biggest outbreaks in South Korea. They've been sampling since the 15th. And so we'll be, again, the sampling is ongoing and the sequencing's just started. So it's really interesting to see what's been around. We're looking in Sao Paulo in Brazil, in Tokyo, of course, in Shibuya Station. And what we've been doing with the samples is, there's a lot of ways you can analyze a sample. So if you could even look at some of the lysis results, but when you get RNA out, we often want to look at, if we can, we'll bank it, but there's fast methods. Some are CRISPR and Cas12a or Cas13 based methods. There's one called LAMP, which is also very fast. I'll show you a little bit of data from that. RT-PCR is what most people are doing today in the clinical labs, what we're doing at Cornell right now. There's also variations of RNA sequencing. I'll let you examine a variety of different features. And also there's direct RNA sequencing like with MinION, and also capture panel. We've been playing with the Twist capture panel, which I'll show you a little bit of data from, and some other controls as well, which are essential to the science to be able to have a control, which I think Twist, and as Emily said, you can brush your teeth with it, which I can confirm it does taste kind of minty. So if you really are running out of toothpaste, you can use the synthetic RNA from Twist. So what we've seen so far is a good coverage. Actually, some of these are clinical positive samples, and also some of the synthetic RNA. So this is the full 30 kb virus. We can see here from some of the first RNA sequencing data from clinical samples, we're seeing good coverage of the virus. We're in the process of uploading these to NextStrain, which I'll show you a slide of shortly, but we can see also some of the controls here you can see are also over here on the left, which are the controls which we know also work great at getting the full length coverage.
Go back a second, Chris, and talk us through the sequencing data here. So this is reverse transcription. So we're looking at DNA now from the RNA virus. Is that right?
No, RNA. So you have to do RT first. So it is basically you have to make cDNA first. You do in order to actually see the virus. And the only way you can do, actually, just to jump back there, the only way you can get the RNA in its naked form is just to do the direct RNA sequencing with MinION. We published a paper in 2012. You could, in principle, do it on PacBio if you put a reverse transcriptase instead of a DNA polymerase in the bottom. But that hasn't been commercialized further. But currently today, if you want to do only the RNA, this is the only way to do so. But once you capture it, you can then actually, as Emily was describing, you can make cDNA, or also was described also from SynBioBeta, you can use that same RT-PCR system, quickly make cDNA, and then you can measure it quantitatively.
Got it. So let's just pause on this technical slide for a second. So you've got these, there are 6 different assays that you are using, or you have used, or that various people around the world are doing. Is that right?
That's just what we're doing. There's other assays that are out there, there's antibody assays, there's variations like I alluded to of the CRISPR methods. This is kind of our current workflow of what we're working on as everything that comes from pathology. And so I think we're viewing this very much as this is probably hopefully a once in a century kind of pandemic. And there's certainly a lot we can learn from these samples. So we're doing everything we can to aliquot and preserve as much of the RNA from all the clinical samples and to examine them as comprehensively as possible. So these methods, we've been using New England Biolabs for the prep kits. There's of course other ones, but this is from Oxford Nanopore. They do direct RNA. And the other good thing about this is you can get full length RNA. You can see here, you can also look at polyadenylation sites. Some of the other sequencing methods look at both host and viral cDNA. You notice here I put cDNA, not RNA, because it's technically what it is. And our RT-PCR is using 2 primers for the envelope and spike genes at the hospital, but there's ongoing questions about which ones you could use other ones as well. And as most of you know, the CDC ones that first came out were actually not that good. It appeared no one had taught them how to use primer 3 at some point. But yeah, so as I think as most people on this call probably use. And then also I'll show you a little bit of data from LAMP, which is a protocol for rapid PCR.
So you have you have the sample collection, then you have the assay that you're using to convert it into a format that you can sequence it. You have the timeline there, and then you have the mechanism that you're using to sequence. So the MinION, that's from Oxford Nanopore, and then you've got a number of other, probably you mentioned Illumina, PacBio. So depending on the method you use, you can use a different set of sequencing technologies on the right there. And then I didn't realize you could do direct RNA sequencing with the Nanopore. Is that right?
That's right. Yeah. So you can, in principle, the only challenge is you need 500 nanograms of polyadenylated RNA. So it's a lot of RNA. The COVID-19 virus, which is the SARS-CoV-2, does have a poly-A tail. So it actually has a 33 nucleotide poly-A tail. So it's why you can do poly-RNA-seq, and you can prime on that with the nanopore sequencer. Another protocol that has been published is you can actually make a splint that actually attaches to what is the motor protein and will only bind to its complementary target. So there's ways you could, in principle, only sequence the SARS-CoV-2 virus as it comes through, but I have not seen it yet. That's something that's been published for other viruses like Zika, and so presumably it's just a design question. But yeah.
Got it. So if you can now go back to the sequence, so the sequence is 30,000 base pairs, if I'm right. If you could just talk us through the sequence and which bits of it are conserved, that is, they're very similar to previous coronaviruses, or they're very similar to other, when you sequence, everything is the same, and talk about which bits are different. And in the different samples, like the Wuhan sample or the Australia sample that Emily mentioned, which bits of those are changing? How much of the variation are we seeing when you do this sequencing of the DNA of all these viruses? Which bits are the same and which bits are different?
Yes, that's a good question. So you can see here some of the colors. Anytime you see a color differential, you will see there are some genetic variants there. And so we can see some of them here. And actually, since I'm sharing my screen, I'll switch quickly over to, I turned off most things, but it's probably the best place to look at all the variants that are known to date is NextStrain. And if people haven't been to this website, it's probably one of my favorite websites to visualize the data. You can see where all the different strains that have emerged over time, and then where essentially do they look like they've been moving. So of course the outbreak started in Wuhan and has since spread quite quickly throughout the world. And you can see, based on the phylogenetic analysis of the nucleotide divergence, where do we see those strains mapping around the world? We've just started uploading some of our data into GIS-8, which is basically where the NextStrain gene pulls that data from and then creates these phylogenetic and basically tracing maps of where do we see the viruses emerging. So you can see, as most of you probably know when you read the news, it's really dramatically expanded in the past few weeks. And is really now truly a global pandemic. At the bottom, you can see what's really exciting is if you look down here, you can change by the number of nucleotides or amino acid changes. Where do we see the most genetic diversity this is, at least from what I've seen so far, probably the best and most straightforward site that displays that. So here you can see the different, in the open reading frames or in some of the basically the envelope gene or the spike gene, you can see where do you see these spikes of genetic diversity. So this is one thing that, of course, Twist thinks about is if you're designing capture probes, you want to design them to be as tolerant as possible in general. But then if you know where the mutations are, you can actually even build probes into that for capture. So, so having these kind of genetic maps helps you design primers, helps you design capture probes, helps you know where the mutations are, and also helps you think about vaccine development, of course.
Chris, this is absolutely amazing. So what we're seeing here is a phylogenetic tree in the top left. So we're seeing how the virus has evolved over time and over geography, and you're seeing different clades emerge as you're showing in the different colors. So a clade being a group of that sequence.
That's right.
Similar viruses with a similar sequence, and then you're being able to track where they came from. So maybe could we just zoom in then on the Seattle, because we know that was the first kind of big outbreak in the US. So can you zoom in on that and just talk a little bit about what we saw in Seattle?
Yeah. And you can see that so this data is usually 1 or 2 days old. It's getting caught up. Well, you can see here, there's actually 1,500 genomes that have been sampled. So you either have to do full-length MinION sequencing or RNA sequencing to get the variant calls for the genomes. And we just finished the first 30 of them here. And we'll have about another 300 by next Wednesday, if all goes well. We'll be uploading our data up here.
Great. And if you want to follow along at home, this is nextstrain.org/ncov, and Kevin can type that into the chat box.
That's
so great. Yeah.
Right, and you can see, if you zoom in, you can see where did this, when it hit Washington, and it kind of puts the little dot, it's not actually in Seattle, it just does it state by state. But you can see when they landed there, and then how big was it, and then where did they go from there? And so, it's an inference, so it's not a perfect map of necessarily, who brought it, but you can see here quite distinct clades. So if you look here, you can see this whole red group here are all the whole genomes that are this distinct genetic subgroup of the virus that almost all look like they're in Seattle, right? So this is, you have to be careful because there's been popularized an L and an S strain based on early divergence, you can see here. But as Trevor Bedford describes, and actually he's one of the programmers here, does brilliant work on this. This could just be how the virus distributed early on. It's not necessarily a functional difference in the virus, but you can see, much like an evolutionary tree, different branches that indicate, potentially different subgroups. But I can't emphasize enough that and these is mostly a European clade you can see here, but occasionally some Australian. And then down here you can see there's a sort of Saudi Arabia sort of samples are here. You see some more in England, and I see some other ones here. So you see variations of them, Taiwan, Hangzhou, some of the Chinese clade also showing up here. So what's cool is you can zoom in if you want to look at each one and just look at specific samples, where did they come from and where did they likely diverge. So it's great.
And if you can just go back to your presentation and show us the sequences, you talked about these spike proteins. We're actually going to be doing a structural analysis of the SARS-CoV-2 virus next Thursday morning at 8 a.m. in a virtual environment called Nanome with some people who are looking at structures. Rob Reinhardt and the founder of Nanome are going to be on the show, as well as Robert Scoble, who's a VR expert, and Philip Rosedale, who's the founder of Second Life. So we're doing a whole bunch of stuff where we're going to bring up the virus shape and let everybody look at it. Tell me about these spike proteins that we keep hearing about, and tell us, tell us what you can about the spike proteins, and then tell us how we're seeing the sequences, how that relates to the sequence we're looking at here.
Yeah, happily. And so if you can see my mouse, this is the spike gene that's down here. And so this is where we see different genetic variants pop up in some of our samples. And we're, again, these will all be up in GSA shortly and also shared with the world for people to help map and understand the pandemic. But there is genetic variation here. And it's, there's other sets of variation as well. But this is what people worry about a lot in the vaccine development, is because this will change a lot of how you can actually design how this virus is read by the cells, is the change in this spike protein. So we do see genetic variants, but it isn't I wouldn't say, I'm not going to call it a hotspot yet, or something quite like that. It seems to be, there is genetic variation, but we see it across other genes as well, and other groups have seen it. So I will not yet make any broad claims, but there is genetic variation, which is very important to know, especially for vaccine development.
Great.
And then just a couple more slides, just to zoom in of some of the variants when you do consensus alignments, where we see, again, this is the reference strain. We can look at some of the clinical samples and also some of the positive controls that we see as well, and some other variants of them. And then I already showed you this. So this is the strain typing data. And our last couple slides are just that we've been actually using this new LAMP assay, which can work in about 30 minutes to see, have we found it in environmental samples? And the answer is so far no. We've done another 100 reactions. And so some of the good news is we haven't yet seen it, any real strong evidence of it in the subway. So that's good. We haven't done extensive sampling, but we're in the middle of all that collection now. And this assay works for clinical samples as well. So we can see it here. And there was a Chinese group that had first tested this out, and we now have it working here as well. And so we'll be sharing those protocols online in a preprint that should be out next week. As a quick side, we've done other pop-up labs before, and I think eventually we could have more pop-up labs like at Biosciences. And quickly, there's a whole bunch of other things you can do once you get sequence data, the end slide here. And the last thing is, what have we found in the subway so far for RNA viruses? So again, no strong evidence of SARS yet, but here's some of the things we do see. Is if you just grab a subway sample and sequence it on the RNA level, which is different from the DNA, we do see about 10% of it is human DNA. We see a lot of it's unknown. So these are cleaned reads, so they should be coming from somewhere. But we have seen flu, actually. We've seen, which is not uncommon at this time of year. We see phage RNAs. We see some other intriguing sort of alpha viruses. There's different viruses there, and some of them, and this is data, this literally came from this morning, so it's very new data. But with anything you find on surfaces, we're doing a lot of double checking to see, if you see tobacco mosaic virus, that might be there from a plant maybe, but you also have to control for the enrichment of how many of these species are in the databases, and make sure you do very careful analysis of the data. So this is very, very new data, but again, we'll be getting thousands of samples over the next few weeks, and we'll have a very comprehensive and multi-omic characterization of them. So stay tuned. There's more to come. And of course, I want to thank everyone, the clinical, academic, government, industry partners. Thanks Twist, who's helping us with the controls very early on, and of course, Krista, Ben, and Councilman Two Bens over there on the right. And that's it. Thanks a lot.
Awesome, Chris. Thank you so much. So the big takeaway, this is a good news webinar. There wasn't SARS-CoV-2 found from your swabbing of the New York subway. There's a whole bunch of other stuff that you found of viruses and influenza and things like that we know are out there. And it really just goes to show the durability of the human body and the human immune system, that all this stuff is out there, and generally we don't need to worry because we're producing antibodies to keep it all out of us. I think the other thing to note from your data is really interesting is that 10% was human and 2% was viral, and that would be 88%.
Unknown, yeah.
It doesn't map to anything. I mean, that's just crazy that after so much sequencing that we're still seeing 88% of the DNA that you find on the New York subway doesn't map to things that we already know about.
Yeah, and that's that's from just a handful of samples. So we're, everyone treat these data as very preliminary, but it's interesting. And also, it's not as crazy as it sounds. Like for the DNA sequencing, even there still, you'll get 50 to 60, sometimes 70% of the fragments don't match any known genome at all. So the fact that our transcriptome is pretty mysterious, or more so in terms of what RNAs are there, is not shocking, but it is higher than what you'd find on the genetic side. So these are very new data. We'll be working on it and sharing. We'll also be sharing these data as soon as we can, so other people can get their eyes on the data and play with it.
Awesome. Well, we have a few minutes left at the end now for Q&A. We have 19 questions asked, so hit the ones that you like and we'll try to get to those. And if you want to ask your question live, raise your hand and I'll dial you in. So if we can go to the panel view and let's ask the biggest question is it still infectious? So that's the big one. Nobody wants to be handling stuff. So Chris, maybe you can take that, and then Josh after that.
Yeah, actually, David Coyle had a great post on this on microbe.net. Is the answer is probably not. So seeing a virus is not the same as it being an infectious virus. Even it being an infectious virus doesn't mean that you'll get infected. Even if you get infected, doesn't mean you'll get sick. So there's all these layers between an actual viral sequence and the manifestation of disease, and using Koch's postulates, for example, to validate them. So what we're looking at is just the presence of the viruses. We are doing testing in Vero 6 cells and other cell culture medium to see, can the virus even infect a cell if it's still there, which is ongoing. So which you have to do to even just see if it is viable. But it's very important to note that just seeing a fragment of RNA on a surface does not mean it's infectious, because there's been reports of up to 9 days or even 17 days of the virus being present on the surface, particularly in the Diamond Princess cruise ship. I don't know if people saw that report, but the virus can hang around for a while, but how long it's actually infectious is something you can only really do in cell culture. You would never get an IRB to say, I'm going to take this subway swab and put it in your mouth, right? So you can do it in cultured cells to test it.
Great. And the IRB is the Institutional Review Board that's approval for doing human studies. Josh, over to you. We have a couple of concerns that you're making these RT-qPCR machines for and the reagents for doing environmental sampling. Isn't that taken away from crucial testing that's needed for real patients? And then also, things like the availability of swabs and things like that. And then we have also a couple of questions for you on the cost per sample and the turnaround time. So Josh, if you can take those ones.
Yeah, I mean, we use our own machines, and we have them available, so we're not concerned that we're taking it away from testing. I mean, people can buy them today. But I'd like to add, with RT-qPCR, it's true that a virus may not be alive, but we're not that concerned about that. We think the greater the thing what we really want is greater sensitivity, and if there's RNA there and it's not alive, but it's detectable, it's well on its way to not being detectable. So I think that it's the greater sensitivity is what's more useful.
Great, excellent. And then we have a question for you, Emily. Are you offering a commercially available package for sequencing environmental samples? And maybe you can also talk about where do people go if they're interested in these panels that you talked about, or ordering them for themselves.
Yeah, absolutely. They are available online. You go to twistbioscience.com/shop, and you can order positive controls. You can order sequencing kits. We provide everything from the sample all the way to being ready to go onto the sequencer, and then you need to have access to an Illumina sequencer to be able to do the analysis. But yeah, it's we try to make it as easy as possible for people to have access, such that we can remove the friction and more testing can be done.
Great, excellent. And we have a couple of interesting questions here. One, looking ahead to a post-pandemic world, a new normal, how do we achieve routine environmental tests at scale, i.e. offices, public spaces, and incorporate that into public health monitoring? I was seeing Balaji, the former CTO of Coinbase, who was online, and he's been talking about creating these green zones and red zones in order to get the economy back to work, where you can have these green zones of people that have been tested, environments that have been tested, and everybody in those green zones can get back to work. Even before you get on a plane, you've got to show what you've, where you've been in the last 14 days, and you can then get on a plane and go to other countries. So what does this post-pandemic world look like, Chris?
Yeah, I think people previously thought that the cost of doing routine surveillance was too expensive. And I think as Josh has shown, and the other kids coming from Twist and what we're doing, it's now actually possible to do things at scale for surveillance. And it's clear that the economic cost of what happens when you don't do it and you haven't caught something in time is probably far worse than whatever the cost would be for even a minimal level of surveillance. So I think the new normal might include a pretty renewed focus on this question.
And then Ellen's asking, have you had resistance from people who don't want you to do environmental testing because they don't want to know the results? Seems quite stupid, but have you had that?
Yeah, so generally most people don't want to know. I prefer to run towards information rather than away from it. But at the end of the day, it's a personal choice. Even what newspaper you pick up will determine what's in your brain a little bit. So you have to make the choice of what you absorb.
Great. And Chris, could you just go into a little bit more detail about this LAMP assay, because it's pretty revolutionary. What does LAMP stand for? And talk about the colorimetric piece of this, because that's one of the innovative parts.
Yeah, and we've been working on that a lot in the past few weeks as a way to get something faster, especially for medical personnel at the hospital. They just, they're on the front lines working all day and, working 16, 17-hour days. So we want to give them a way to quickly test if they might be positive, at least as a screen. And so it stands for loop-mediated isothermal amplification. It was first published by a Japanese group in 2014 and commercialized by New England Biolabs that same year. And now it's available as a kit. You just have to make sure you get the right primers and you can optimize the reaction. But it is something that is pretty, we think, intriguing and has tested a bit in China. And we're also, again, it's going to be in the preprint that's coming out soon. So stay tuned. And we have a lot of data on that. But it looks as a really promising way forward for sure.
Great. Excellent. Well, we're coming up towards the end of the town hall today. Thank you all. We haven't got around to all of the questions, but it's certainly been a very interactive one. Keith Robertson has a question. Any estimate of LOD of your sequencing approach? Maybe you can just define LOD for us, Chris.
Yes, from the samples we've done on the sequencing, if you do a twist capture, it in theory goes down to one virus. Because that's the advantage of capture, is you get rid of everything else. We've just been looking at the data now for we think the sensitivity looks to be about 90%. It actually correlates very well with the RT-PCR. If you have a high viral titer, you get a lot of the RNA. It's very easy to see in RNA sequencing. If it's a low viral copy, you have to either do PCR to enrich for it or capture to get it out. Otherwise, you just won't see it.
Excellent. Well, just enough time for me to thank all of our panelists today. Emily Leproust, the CEO of Twist Biosciences, Josh Perfetto, the CEO of Chai Bio, and Chris Mason, professor at Cornell University. We hope that you enjoyed this. Don't panic. The virus isn't everywhere, and stay socially isolated, stay happy, stay safe, and may all your Zooms today have space station backgrounds. Thank you all for joining us for today's SynBioBeta Live. A lot of fun.
Thanks, John.
Thank you, John.
Thanks, everyone. We'll see you soon.
Bye.
Okay. Cheers. Bye-bye now.